Welcome to Animal Biotechnology. Today, we are diving into the fascinating world of viral vectors, specifically the Lambda and M13 bacteriophages. Imagine trying to deliver a fragile package into a heavily guarded fortress. You need a specialized delivery truck. In molecular biology, these viruses act as our microscopic delivery trucks, allowing us to safely and efficiently transport genetic material into bacterial cells. Let us explore how they work. Now that we understand the basic premise of a biological delivery truck, let us look at our first vehicle, the Lambda Bacteriophage. In nature, bacteriophages are highly evolved microscopic machines designed to inject their genetic material into bacteria. By carefully removing the harmful viral genes and replacing them with our own DNA of interest, we can hijack this natural delivery system. This makes the Lambda bacteriophage an exceptional tool for genetic engineers who need to move large pieces of DNA into cells safely and efficiently, acting much like a biological syringe. Building on this idea of a biological syringe, let us examine what exactly is inside the Lambda bacteriophage. It is a temperate virus that naturally infects E. coli bacteria. Its natural genome consists of a linear, double-stranded DNA molecule that is about 48.5 kilobases in length. Interestingly, at each end of this linear DNA, there are single-stranded overhangs of 12 nucleotides. These are known as cohesive ends, or COS sites. These COS sites are crucial because they allow the linear DNA to circularize immediately once it enters the host cell, protecting it from degradation. Knowing how the genome is structured leads us to how we actually use it in the lab. To use Lambda as a cloning vector, we genetically modify it to remove non-essential genes, creating physical space for foreign DNA. We use two main types: insertion vectors, which simply open up to accept a small piece of DNA, and replacement vectors, where a central stuffer fragment is swapped out for a much larger foreign DNA fragment. Why use Lambda instead of standard plasmids? Plasmids become highly unstable when inserting DNA larger than 10 kilobases. Lambda naturally accommodates massive inserts between 10 and 25 kilobases, which is absolutely crucial for constructing large genomic libraries. This high capacity we just discussed is not infinite. It is strictly governed by the physical geometry of the viral protein shell. The packaging machinery requires the total DNA length to be within a specific range. We can express this maximum insert size mathematically. The formula states that Imax equals Pmax minus V essential. Here, Imax represents the maximum size of the foreign DNA insert we can add, measured in kilobases. Pmax stands for the maximum packaging capacity of the phage head, typically around 52 kilobases. Finally, V essential represents the size of the essential viral DNA that must remain for the virus to function. Physically, if we engineer a vector with less essential DNA, the available room for our custom insert increases proportionally. Before we move on to our next virus, let us see this formula in action with a concrete example. A genetic engineer is using a Lambda replacement vector to build a genomic library. We are given that the maximum packaging limit of the phage head is 52 kilobases, and the essential left and right arms of the vector total 28 kilobases. We need to find the maximum size of the foreign DNA fragment that can be cloned. We chose our packaging limit formula because it directly relates these variables. By substituting 52 for the maximum capacity and 28 for the essential DNA, we perform a straightforward subtraction. The result is 24. This means the engineer can successfully clone a continuous piece of foreign DNA up to 24,000 base pairs long into this specific vector without bursting the phage head. Now that we have mastered the heavy-duty delivery of the lambda phage, let us shift our focus to a completely different type of viral vector, the M13 bacteriophage. Think of a standard virus as a bomb that explodes the cell to release new viruses. M13, however, acts more like a gentle parasite or a continuous factory. It enters the cell, sets up a small assembly line, and constantly secretes new viruses through the cell wall without ever killing the host bacterium. This unique behavior allows the bacteria to survive and continuously produce the exact DNA we want, making M13 an incredibly sustainable and efficient biological factory for genetic engineers. To understand how this biological factory operates, we need to look at its life cycle. M13 is a filamentous bacteriophage that specifically targets E. coli cells bearing an F. pilus, a specific bacterial appendage. Unlike Lambda, which carries double-stranded DNA, the infectious M13 particle contains a circular, single-stranded DNA genome. Once inside the host bacterium, host enzymes convert this single strand into a double-stranded replicative form. This replicative form is brilliant because it acts just like a standard plasmid, allowing us to easily insert foreign DNA using standard restriction enzymes before the virus starts pumping out new single-stranded copies. Let us visualize this incredible continuous factory process we just discussed. On screen, you are looking at a schematic diagram of the M13 phage life cycle. On the far left, you can see the filamentous M13 phage attaching to the F. pilus of the E. coli bacterium. As we move inside the cell, the circular single-stranded DNA is shown converting into the double-stranded replicative form. Notice the cycle in the middle. This double-stranded form is used to synthesize brand new circular single-stranded DNA. Finally, on the right side, you see these new single strands being packaged into protein coats and extruding through the bacterial cell wall. The key takeaway here is the non-lytic release. The cell remains completely intact and alive while producing our DNA. Seeing how M113 produces single-stranded DNA brings us to an important question. Why do we even need single-stranded DNA in biotechnology? While double-stranded DNA is great for storing genes, many advanced molecular biology techniques specifically require a single strand as a template. For example, the traditional Sanger DNA sequencing method requires a single-stranded template to read the genetic code accurately without any interference from a complementary strand. Additionally, site-directed mutagenesis, which we use to intentionally change specific amino acids in a protein, relies heavily on single-stranded DNA to bind mutated primers. M13 vectors provide a pure, endless supply of this material. To efficiently run this single-stranded DNA factory, we must carefully control the infection process. When working with M13 phages, researchers need to know exactly how many virus particles to add to their bacterial culture to ensure every bacterium gets infected without wasting resources. This ratio is called the multiplicity of infection, and we calculate it using a simple formula. M equals P divided by B. Here, M stands for the multiplicity of infection, which is a unitless ratio. P represents the total number of infectious phage particles added, and B represents the total number of bacterial cells present in the culture. Physically, if the number of phage particles increases while the bacteria count stays the same, the ratio increases, meaning multiple phages might attack a single bacterium. Now that we have our formula, let us apply it to a real laboratory scenario. A researcher is preparing an M13 infection to produce single-stranded DNA for a sequencing project. We are given a liquid culture containing 2 × 10⁸ E. coli cells and a stock solution containing 1 × 10⁹ phage particles. We need to find the multiplicity of infection. We use our ratio formula because it tells us the average number of phages targeting each cell. By substituting our values, we divide 1 billion phages by 200 million bacteria. The math simplifies beautifully to give us a final answer of 5. This means, on average, there are 5 M13 phage particles for every single E. coli cell, which is an excellent ratio to maximize our DNA yield. We have covered a lot of ground today, so let us summarize the most important ideas from this topic. We explored 2 highly specialized viral vectors that serve completely different yet equally vital purposes. First, we saw that the Lambda Bacteriophage acts as a heavy-duty cargo ship, carrying massive double-stranded DNA fragments that are absolutely necessary for building comprehensive genomic libraries. Then, we learned that the M13 bacteriophage acts as a specialized biological factory, continuously pumping out pure single-stranded DNA needed for sequencing and mutagenesis. Together, these 2 vectors expand the genetic engineer's toolkit far beyond what simple plasmids can achieve. You now have a solid understanding of how we manipulate animal and human genomes using these incredible microscopic machines.